Okay. So when we left off, we were talking about metabolism. So we're going to jump right back into that topic. Oh, and I'm going to let people in. Okay, cool. Okay. So up until this point, we've been talking about two types of substrate level phosphorylation. So we talked about glycolysis. We talked about the fact that that takes us from glucose to 2 molecules of pyruvate, produces 2 net ATP along the way. That pyruvate moves into the mitochondria where we have the Krebs cycle, which again produces 2 molecules of ATP, 1 per cycle. But we know that metabolism as a whole, our aerobic cellular respiration, creates a lot more than 4 ATP molecules, right? So the bulk of this ATP is going to be formed by the electron transport chain in the mitochondria. So what's happening is that electrons are going to move down the electron transport chain, and that process is ultimately going to synthesize ATP. This process is where we need to use oxygen. So that's why this is aerobic cellular respiration. Okay, so the electron transport chain. Okay, so the electron transport chain is embedded in the inner membrane of the mitochondria. So there are these chain of molecules, these chain of proteins in that membrane embedded in it. So we've been taking all these reduced electron carriers. We've been reducing NAD+ to NADH. Now we're going to move them to the electron transport chain where we're going to free those electrons, move them down the electron transport chain in order to harness that energy. This is the process of oxidative phosphorylation. So as we release that energy to make a big boom of ATP, that is a different process than the substrate level phosphorylation we've been doing up until now. And this is going to require the molecule ATP synthase, which we're also going to see in that mitochondrial membrane. So our last electron acceptor at the end of this whole process is going to be O2, so it's going to be oxygen. So this is where we need oxygen just right at the very end. So it almost seems like a little aside, but if we don't have that last electron acceptor, if we don't have that oxygen, everything gets backed up. We can't sort of release this gradient that we're trying to build. So we actually can't use the electron transport chain at all if we don't have oxygen present. Okay. So here's what the electron transport chain looks like in terms of the formula. All right, so we're donating electrons to this chain. So we've been adding electrons on to our NAD+ to reduce it to NADH. Now we're going to be peeling off those hydrogens and peeling off the electrons we've been adding on. Finally, we add in those electrons and some hydrogen ions to that oxygen, and we're forming water as a byproduct. So this is why in our full reaction for glucose metabolism, we see that water at the end. Right, so this is where we're producing a lot of that water. So we want to think about, for the purposes of energy creation, we want to now realize why we cared about those reduced electron carriers. It's because we need them for this process, and this is the process where we're making so much energy compared to glycolysis and compared to the Krebs cycle. Okay. Sorry, my buttons are different now that I'm on here. All right, well, we'll just have to look for now. Okay. Well, what we see happening right here, so we're looking at the structure of the mitochondria on the left. So that red structure, we see that it's outer layer here in red, that's the outer membrane. Then we see a space here before we see that inner squiggly orange membrane. This is the intermembrane space. So that space is really important because as we're peeling off these electrons from our reduced electron carriers, we're basically pumping a bunch of hydrogen ions into that space. So we're creating a gradient where we have a lot of hydrogen ions, which we can also think of as protons. So a bunch of hydrogen ions here in the intermembrane space, and not very many left at all inside the very center of the mitochondria, which we call the matrix. So we're building up a gradient. And when we think about gradients across membranes, we can almost think about that gradient as a form of energy, because as we'll see, as we start to talk about movement of molecules across membranes, molecules basically want to move down a gradient. They want to spread out so they're evenly spaced. So they want to move from high concentrations to low concentrations to get more room. So as we're creating this separation between an area where we have a really high concentration and an area where we have a really low concentration, we're essentially building up this desire to move. So we see these first three coenzymes, right? These are proteins embedded in that inner membrane, the orange one. And here we see the ATP synthase at the end. And so that ATP synthase is where we're going to have all of these hydrogen ions rush through. It basically spins. And as it spins, it turns our ADP, our adenosine diphosphate, into ATP, adenosine triphosphate. So the role of these coenzymes earlier on is to pump those hydrogen ions out into that intermembrane space, building up this gradient, and then ATP releases that gradient. Now, a thing to notice, I'm just going to point out about this diagram because we'll see it again, this kind of color scheme. So as we start to talk about types of transport, you'll notice that these arrows in those coenzymes are red. That means that's a form of active transport, so it's using energy. And then you'll see that going through ATP synthase, rushing into the cell, that's green. That means it's going to be a form of passive transport. It's going to be favorable. It's not going to use energy. And in this case, it's actually harnessing that energy and putting it into the form of ATP, creating that final bond. So here we just see that zoomed in again. I believe we're going to progress through. Yeah. So here we see this first step. So we have our NADH coming in plus a hydrogen ion. And we see we're peeling off those electrons, peeling off that H, that hydrogen at the end of the NADH as well. And you can see from that red arrow, we're pushing one of those little blue hydrogen ions across. As we move down the chain, we keep doing things like that. At the end here, so in our final embedded membrane protein before we get to the ATP synthase, we can see that we take those electrons at the end, and our final electron acceptor, rather than being one of the membrane proteins, is oxygen, giving us water as a byproduct. Finally, we release that gradient and allow our cell to form ATP from ADP using this energy. So after this, that gradient would get released. Basically, we start to see equilibrium state, so equally balanced hydrogen ions on either side of that membrane. And then it would begin to build up again as we want to produce more energy. So when we're looking at our whole process, it's going to look like this. Okay, so we started up at the top with glycolysis. So in our glycolysis step, we're out in the cytoplasm. We put in some starter molecules. We put in our glucose, that's what we're breaking down, but we also put in some NAD+, some ADP, and on net, We got 2 molecules of ATP and a couple, 2 molecules of NADH plus some hydrogen ions. Our carbon is now in the form of pyruvate, which is going to move into the mitochondria. It's going to join up with some CoA to form this acetyl-CoA. In this process, it's creating some more reduced electron carriers. And we're also giving off some carbon dioxide as we peel off a carbon from each of those pyruvate molecules and go down from a 3-carbon molecule to a 2-carbon molecule. The acetyl-CoA goes into the Krebs cycle. And then we go around and around. All right, so we're going around and around. Each cycle, which we have 2 cycles because we had 2 pyruvate molecules, 2 acetyl-CoA, produces 1 ATP. So on net per 1 molecule of glucose, we've created 2 ATP. What we notice we make an awful lot of in the Krebs cycle is those reduced electron carriers. So that's our 6 NADH. And that's what I would consider the most important point of the Krebs cycle, okay? Because we're sending all of these reduced electron carriers to the end, to the electron transport chain to go into oxidative phosphorylation, where we make on the order of 28 molecules of ATP. So this is our whole sort of trace out process for aerobic respiration. So in the presence of oxygen, our glucose oxidation. So this runs us out, our glycolysis, creating our pyruvate, creating acetyl-CoA, going into the Krebs cycle, and then into oxidative phosphorylation. So this is how we get our overall reaction. So we started with the glucose up the top. Okay. And we made 32 to 38 ATP molecules coming from our glycolysis and our Krebs cycle, as well as this oxidative phosphorylation step for which we use the electron transport chain. And we had some byproducts. So we actually don't have any carbon left at the end. We pulled out all of those 6 atoms of carbon, release them as carbon dioxide. That's why you breathe off carbon dioxide. We also created water at the end there as we have the oxygen accepting those final electrons. Questions about this overall process?
What determines like 32 to 38?
So it's the gradient that's forming across that inner mitochondrial membrane. So essentially, the more of a gradient you can build up, the more ATP you're going to be able to make, like the more power you have to kind of force stuff through and spin the ATP synthase. So that inner membrane is kind of variably leaky. It mostly doesn't let the hydrogen ions through. I think there's a section somewhere in your book about variation here. In different types of tissue and people of different ages, some can kind of leak back across. So that gradient doesn't build up as big. And so then you don't get as much of a bang for your buck at the end, basically. You want to know the steps? Yeah. Yep. You want to be able to talk about whether they make ATP, the reduced electron carriers. Mainly, first thing to focus on is the ATP. That's your like first order of business. So I keep mentioning it. Yeah.
Do we have to know the specific numbers for like how much NAD plus is made or something like that, or we'll just know the overall process?
No, the overall process and the number of ATP, but
Okay.
the NADH, don't worry
Okay.
about that so much. Yeah. Okay. So next we're going to talk about what happens if we don't have the oxygen. Because I said that we have that backup, right? If the electron transport chain is running, we don't have oxygen at the end, that whole process doesn't work. We kind of can't release stuff without having that electron accepted by oxygen. Your cell is still going to want to make energy though, right? So if you're going on like a long run or lifting really hard, think about your muscle tissue, right? It might be using up all the oxygen to form as much ATP as possible, but sometimes it's doing it too fast. There are also organisms that live in environments that are low in oxygen, but for the purposes of A and P, we're really thinking about tissues that are deprived of oxygen. So if we have low oxygen availability, this means that our electron transport chain is backing up, and the Krebs cycle, which is also happening in the matrix in that center of the mitochondria, it stops spinning. So we can't do these final two processes. So we can't run that final step where we're getting on the order of 28-ish ATP. And we also aren't getting our too little ATP from the Krebs cycle. But we do still want energy. So the way we're going to do this is by running glycolysis. So glycolysis was that step out in the cytoplasm before we moved pyruvate into the mitochondria. So we don't really need mitochondria at all for this step, actually. We can do that, but we can only do that if we have NAD plus available, because part of that process of glycolysis was reducing that electron carrier. So now that NADH that we've been making through glycolysis, right, it has nowhere to go anymore, right? We were sending it into the mitochondria to the electron transport chain, but if the electron transport chain is all backed up, it can't do it, it can't accept it, and it can't turn it back into NAD+. And if we don't have that NAD+, we can't run glycolysis. So this poses a problem for us. And this is where we're going to see the role of lactate. So we're coming up with like an alternate method of turning that NADH back into NAD+. So we can keep running glycolysis and keep getting over and over and over again our two little molecules of ATP. So it's not as efficient as if we have oxygen, but it will give us some energy so long as we have the molecules to feed into it to do it. Okay. So here's what that looks like. So we're still starting out with our glucose molecule. So that's the same as normal. We're still going into glycolysis, right? So we're still taking that glucose molecule, using a bunch of enzymes and breaking it down and making pyruvate. So all of that part is the same. So that carbon part. We still need to feed in NAD+, and we are still going to be making NADH. So again, that's the same. And we're still making on net 2 ATP. So all of those steps are the same. So if we wrote out an equation for glycolysis in this process, it would actually be exactly the same. What's different is what's happening to that pyruvate. Okay. So we'll see that the pyruvate, before we were taking that, turning it into our acetyl-CoA molecules and putting those into the Krebs cycle. We're not doing that anymore. The Krebs cycle isn't working anymore. The electron transport chain isn't working anymore. However, we still need this NAD+ for glycolysis to run, because our equation for glycolysis is exactly the same as it was before, regardless of the fact that we don't really need electron carriers. So what we're going to do is we're going to use an enzyme lactate dehydrogenase. Okay. And what lactate dehydrogenase does is it turns pyruvate into this molecule lactate. And that process happens to use NADH, oxidize it, turn it back into NAD+. Right, so that's very convenient for us. And that's really why we turn pyruvate into lactate, is just to peel that electron off of that reduced electron carrier so that we can keep running glycolysis. So if you've ever heard of lactic acid, and here it's not actually in that form in the body usually, it's in the form of lactate more often. But this is what's going on, is that as we're using our body, as we're running out of oxygen because we're using a lot of energy, we want to keep running glycolysis even though the electron transport chain is getting backed up. And the way we can run glycolysis is if we have all these precursor molecules, including our oxidized form of our electron carrier, that NAD+. And the way we can get that is turning pyruvate into lactate instead of turning it into that acetyl-CoA. Questions about this overall process? It's really just a sideways way to get that NADH to turn back into NAD+ in the absence of oxygen. Let's see if this is clear. It's not showing on the screen. Okay. Yeah. So next, we're going to move on to how we break down other molecules. So we've talked about ATP, right? So we know that ATP is where we're storing our energy as we break down the bonds in glucose. We know that we're getting some of it from glycolysis, 2 molecules on net. We're getting 2 from the Krebs cycle if we have oxygen. Then we're getting on the order of 28-ish from oxidative phosphorylation done by the electron transport chain. So we talked about the different events there, so our different steps. Okay. Now we're thinking about other things we can break down. So we're not just always eating plain sugar monomers, and glucose is a very specific sugar monomer also, right? So we eat fats, we eat protein, our body stores glucose in the form of glycogen. So we also want to be able to use those for energy. So we're going to start out by thinking about our carbs, our lipids, and our proteins, right? Because these are good ways of storing energy as well. Fundamentally, what we're going to be doing is kind of hijacking different parts of this process in order to break down other molecules. So if, say, you eat a protein, right? So we know our protein is our chain of amino acids built into a polypeptide, forming those alpha and beta sheets, crumpling up as the R groups interact in that tertiary structure, and potentially building on with another subunit. We can break those down to their constituent amino acids. So we can break that chain down, peel off those little amino acids through our digestive system. And when we do that, we can actually feed those amino acids, not into glycolysis, but into later parts of this process. So we can turn some of those amino acids into pyruvate. And then the process from there would just run as normal. So we'd turn an amino acid into pyruvate, turn it into acetyl-CoA, go through the Krebs cycle, go through oxidative phosphorylation. We might turn it into acetyl-CoA more directly, or we might send it into a different molecule going into a different part of the Krebs cycle. Those are all things that could happen as we break down this protein, depending what amino acids we're peeling off. With our fats, our triglycerides specifically, we're going to break off glycerol from those 3 fatty acids. So we know that a triglyceride, a fat or an oil, is a glycerol backbone with 3 fatty acids hanging off. The glycerol would actually go into glycolysis and use some of those same enzymes, and then we would just proceed as normal, right, all the way through. The fatty acids are going to get pulled into these later parts of the cycle. So they're going to get pulled into the acetyl-CoA, go through the Krebs cycle. They're also going to get pulled into this final step. And when we think about glycogen, so glycogen is a storage form for glucose in animal cells, including us, right? So glycogen, what we're going to need to do is we're going to need to pull those glucose molecules apart, and then we'll have glucose and we can just run this system as normal. So when we pull glycogen apart, that's called glycogenolysis. Glyco for sugar, for that glucose, or geno, we could read that whole thing, glycogen, and then lysis for breaking something down. And I'm breaking down that word for you because we're going to see a lot of similar looking words, right? So probably easier, glycogen and lysis, right? Breaking glycogen. We're also going to see that we can do the opposite process in certain conditions. If we have a lot of glucose, basically we might want to store that glucose in the form of glycogen. So we're also going to have glycogenesis, where we create glycogen and wait until we need more glucose, till we're really active, till we need all that ATP. Okay. So glycogen metabolism is going to be that breakdown of glycogen. So glycogen we find mainly in the liver, glycogen in the liver, a little bit in the kidney. We're going to be storing up glucose for later use. And it's not going to be used just by the liver, just by the kidney. We're going to break it apart, and then we're going to send that glucose into the blood to our other tissues that are looking for glucose to make ATP to run their processes. Now, glucose can leave a cell, but as we break apart glycogen, that's actually not the first thing we get. As we break apart that glycogen, we're going to see this molecule, glucose 6-phosphate. Glucose 6-phosphate can be converted into glucose, but only the liver and the kidney have the necessary enzyme to do that. So that's why we're seeing glycogen pulled apart in the liver and the kidneys, because they have that enzyme that's going to be able to turn it into glucose and migrate it into the blood. So we have this storage form, glycogen. So we're going to want to be able to break it apart, and we're going to want to be able to make glycogen as well. Or sorry, we're going to want to make glucose from a bunch of places. Sorry, I misread that word too. We have a lot of similar looking words. Okay, so we were talking on the previous side about glycogen metabolism. So we can break glucose off the glycogen. Another thing we can do is make brand new glucose. So when we make brand new glucose, this is also happening in the liver, but it's a different process than the glycogenolysis. So what we're going to do is we're going to take glycerol, lactate, and some amino acids, and we're going to be building those into a glucose molecule. So it is going to be different than glycolysis, because there are going to be other enzymes we need. Specifically, we are going to need the glucose 6-phosphatase enzyme. All right. So here we see a map of the different things that can happen with this glucose 6-phosphate molecule and with glycogen. All right. So the part that I find most obvious, right, is that we can take the glycogen, we can break it apart, so we can lyse it. Glycogenolysis becomes glucose 6-phosphate. Okay. And then that glucose 6-phosphate can be converted by glucose 6-phosphatase into glucose, which we can then use. So that whole process that I just traced out in blue is what would be happening when we do this glycogenolysis. Okay. But we can see other things on this map as well. If we trace it from glucose, we can see that we could take glucose and this other enzyme. So when we see that ACE, we know we're thinking about an enzyme. So glucose with our hexokinase can make our glucose 6-phosphate. And then we could create glycogen. So that's our glycogenesis here. Yeah. Oh, so the other thing we could do is take that pyruvate. Pyruvate we could turn into glucose 6-phosphate, And we could also turn that into glucose. So that was that final one, the gluconeogenesis. Okay. Now, long-term energy storage actually doesn't happen in the form of glycogen. So we have a limited amount of glycogen stored in the liver and the kidney that we can use semi-immediately. Your body's first going to want to use glucose if it's available, and it's going to break down its glycogen stores. But most of your energy in your body is stored in the form of fat. It's stored in the form of triglycerides. That's a longer term storage method. So we absolutely do need to be able to break down those triglycerides. So we remember that our triglyceride, it's kind of this flag structure. So first we have that glycerol backbone, and then we have our hydrocarbon chains, those 3 fatty acids. Okay. They look kind of squiggly. Lipids, including these triglycerides, have a lot more energy per weight than carbs and proteins, because we're going to be able to break down those fatty acid chains in chunks. And each little chunk of the fatty acid is going to go into our process and give us energy. So we have energy packed really tight into those hydrocarbon chains, actually. Now, the reason we think about fat in our body as like fat is that those triglycerides, these fats and oils, these molecules are going to be stored in fat cells. So adipose tissue is adipocytes, those are the fat cells. So these triglycerides would be in those cells. So they are stored physically in a cell. Okay. So as we have fat metabolism, first thing we're doing is we're peeling off our backbone, our glycerol, from our fatty acids. So glycerol is basically a carbohydrate. It's going to go into glycolysis. Just do that kind of normal, same process as glucose did. But the fatty acids are something different. So we're going to trace them separately. So those fatty acids are undergoing something called beta oxidation. So they're doing this just 2 carbons at a time, right? So just a reminder about as we draw chemicals, right? So when I draw this squiggly little thing, right, like each point is a carbon molecule with hydrogens coming off of it, right? That's what this represents. We're just lazy when we're drawing them, right? Because we all know, or at least now we all know that that's what that means. So what we're doing when we do beta oxidation is we're taking off 2 carbons with their hydrogens bonded on, and every single 2 carbons little set in that chain is going to give us acetyl-CoA, NADH, and FADH. So we got some electron carriers and we're making acetyl-CoA. And we know that acetyl-CoA goes into the Krebs cycle. Those NADH and FADH go into the electron transport chain. And then we feed into that final step of oxidative phosphorylation. So we're not going to get as much ATP, like we're not going to get that full 32 to 38, whatever sort of number we choose at the end there, depending on tissue type at the end, because we're missing the glycolysis step. We don't produce quite as much NADH. We don't have quite as many reduced electron carriers when we're just taking 2 carbons with their little hydrogens in beta oxidation. So we're going to get slightly less ATP from every set of 2 little carbons. We're only going to get 17. So that question about why we get variable amounts with the electron transport chain was a great one here, right? We're not building up as strong of a gradient. We're not getting as much of a gradient. So we get slightly less ATP. And we also skipped kind of that glycolysis step. But the reason we say that fats have much more ability to store energy, despite the fact that we're only getting 17 for each 2 carbons, is that those fatty acid chains, they are definitely not just 2 carbons long. So it's 17 plus 17 plus 17 plus 17 for every set of 2. So that's what's going on with those fatty acids. Up there, we have a section on the glycerol, but I would consider what's going on with the glycerol less important. We are still using the glycerol. We're trying to use everything we can for energy, but that part is just going into glycolysis. We're going to make slight changes to that process. We're going to build up slightly less of a gradient. So that process with the glycerol is only giving us 20 ATP. But overall, because we're adding all these things together, a triglyceride is going to give us way, way, way more energy than like one molecule of glucose. So here we're pulling out the proteins. So if we want to refer to the process of breaking down a protein, we're going to call it proteo or protein. Lysis, we're still breaking it down. So we're breaking those proteins down into their amino acids. So those individual building blocks, those monomers of the protein. We are going to deaminate these amino acids. So we're going to take our amino acid and we're going to remove the amino group. So remember, the amino group was at one end of our amino acid. We had our carboxyl here. Here was the amine group. Something like that. So we're deaminating, removing that. And that's going to leave us with a keto acid and some ammonia. That keto acid is going to give us pyruvate or acetyl-CoA, which is going to go into the Krebs cycle. All right. So we're going to move directly for a couple minutes into membrane transport. So I want you to take like 1 minute and draw whatever you can remember about a plasma membrane. Hopefully you have looked at cells before and their structure. We're going to go over a little bit about the membrane properties so that next time we can go into transport. We're going to be spending the rest of the time until our exam in different types of transport, essentially. So as we go through our membranes, we're going to be thinking really especially about our phospholipids. So one of our special groups within our lipids that we talked about, that one with the amphipathic properties, because they're going to be our building block of our plasma membrane. That's the bulk of the membrane of the cell. We're going to be thinking about that. And ultimately, we're going to be talking about gradients. We're going to be talking about the chemical gradients, which is probably what you focused most on in your previous courses. So what we're going to be adding on new as we talk about transport are our electrical gradients, and the fact that the electrical gradient and the chemical gradient for any molecule that has a charge is going to add together to form something called the electrochemical driving force. And it's actually that electrochemical driving force that we're going to be paying attention to determine whether we have passive transport, so something that's energetically favorable, or something that's active transport, something that we have to really work at to push something out of a cell or pull it in using energy. Okay. So as we're talking about a plasma membrane, we talk about something called the fluid mosaic model, which is to say the membrane is fluid, it moves around, we can poke at it, it jiggles. It's a mosaic, it's made up of a lot of different parts. So we're going to have different little pieces kind of all nested together to form our plasma membrane. So we're going to have phospholipids as the biggest part. Right? So we're going to have our phospholipids. We're going to have cholesterol embedded in that fluid mosaic model in that membrane because we're talking about an animal cell. So cholesterol is going to kind of control that fluidity a little bit, give us some structure. We're going to talk an awful lot about membrane proteins. If, as we're going through this course, we're talking about something in a membrane, we don't really specify what type of biomolecule it is almost certainly a protein. So when we talk about receptors, we're going to be talking about membrane proteins. When we talk about pumps, we're talking about membrane proteins and so on. We're also going to have a couple of membrane carbohydrates. So we're going to have a little bit of carbohydrates sticking off essentially our membrane across this mosaic, which is sometimes important for like cell signaling, responses of cells to each other. We won't focus as much on that in this course, but you may see it again in the future. So we're going to start with those phospholipids. So those phospholipids are a basic structure of the membrane. And they're the most important part to get down in terms of their like chemical and physical properties, because the presence of these phospholipids, the fact that that's the bulk of our membrane is going to control what can cross the membrane and what can't. So when we were talking about our fluid compartments, we started by breaking down the ICF and the ECF. So the intracellular fluid, anything inside the cell, so the cytosol, the cytoplasm, and the ECF, that extracellular fluid, anywhere outside of the cell, right? We talked about those as part of our divisions of total body water because they are mainly water, right? So our phospholipids, because they have these fatty acid chains that face each other in the center, and we'll trace them out in a second, are going to be a barrier to most water-soluble substances because water is a polar molecule, means things with little charges are going to be attracted to that water. They're going to be repelled by the fact that those fatty acid chains in the center are nonpolar or hydrophobic. Phospholipids also basically jiggle against each other, so they add to the fluidity of that membrane. Right? They are impermeable to water. So we talk about cells as allowing water to cross. We talk about osmosis. There are actually specific membrane proteins there that are specifically to allow water to cross. We won't really talk about them until next semester when we talk about aquaporins as part of our kidneys, but they are there, they do exist. And we'll see the different hormones that are involved with kind of controlling where they are and occasionally where they aren't. So that's why we talk about membranes as being permeable to water, is because of the presence of those aquaporins. So our phospholipids, we're going to think about our cell. We're drawing it as a circle. Remember, it's actually a sphere. And I'm just going to pull out this little chunk here in the gap and draw on the phospholipids that would be here. So we have a bilayer, right? Which means we have our polar head, that phosphate polar head of the phospholipid, facing anywhere we have water. So it's facing the extracellular fluid, and it's also facing the intracellular fluid because we have water in the cytosol. which means that at the opposite end, we have those fatty acid tails facing the center part of this bilayer. So facing each other, they're like foot to foot here on our plasma membrane. And that's how they're forming this structure. So we'll pick up here next time and talk about what's embedded in that membrane.